So good evening, everyone. So today we will be discussing about bioethanol and biobutanol production. Also, the characteristics of bioethanol and biobutanol. So we know that these two fuels, which are derived from the biomass through various conversion technology. Especially, we will be focusing on the fermentation and microbiological conversion processes. Through the production of this bioethanol and biobutanol, we are in a trending way to replace the fossil fuels, diesel, and gasoline consumption in the diesel engines. So, starting with first, we will be discussing on the bioethanol part. So, we know that since 1980s, ethanol has been considered one of the possible and very potential alternative fuels in many of the countries. Now, ethanol, we are considering the bioethanol, which is nothing but ethanol which has been derived from the biomass sources, that is C2H5OH. It is a liquid biofuel which are being derived from various biomass feedstock using various conversion technologies. So this bioethanol is an attractive alternative fuels because it is renewable in the bio-based resources, and as it is oxygenated, thereby it has lot of potential to reduce the particulate emissions in the compression ignition engine, IC engine. So this bioethanol is very appropriate for mixing with the gasoline in the gasoline engine because the self ignition property of the uh, bioethanol is being promoted while it is blending with the diesel and bioethanol in a mixture way. So there is popular blend that usually being practiced in light duty vehicles, that is E85. That means there will be 85% bioethanol and rest of the 15% will be gasoline. But as this bioethanol production still is in cost competitive in the market, so many of the countries they follow the E10 method, that is the 10% bioethanol mixing with the rest of the gasoline will be there. They follow the E10 method. Okay, so the potential bioethanol production could replace around 353 billion liters of gasoline, which is almost 32% of the global gasoline consumption when bioethanol is used in E85 fuel for mid-sized passenger vehicles. So you can also see that bioethanol has huge potential in the trending of as a alternative fuel, right? Because they are the gasoline property and the bioethanol property are very similar and somewhere better in the form of controlling the emission from the vehicle's engine, right? So this bioethanol, which has higher octane number due to the higher oxygen content, it has also the broader flammability limits, higher flame speeds, and higher heat of vaporization than the gasoline. So these all properties allow for higher compression ratio, shorter burning time, and leaner burn engine, which leads to theoretical efficiency which is advantageous over the gasoline in kind of IC engine. It has some sort of disadvantage too, like it has lower energy density than the gasoline. So like bioethanol has 66% of the energy that gasoline has. Okay, so it has lower energy density. It also, its nature is corrosive, low flame luminosity, and low vapor pressure, which makes a cold start very difficult. and it is also miscible with water. So this kind of disadvantage it has, but moreover we are focusing on the advantageous part of the bioethanol over the gasoline. So in all over the world, many of the countries have taken this practice to blend the bioethanol with the existing diesel fuels. So when we are considering the bioethanol production, there are lot of generations of the feedstock. Earlier also we discussed the biofuel generation wise that there are four uh, fourth generation up to fourth generation of the biofuel we can produce depending on the variation of different feedstock. So uh, that is the that biofuel which is originated from various organic waste or sugar beet cereal corns rice and those kind of thing through their processing of the biomass. Now this biological feedstock that contain appreciable amount of the sugar which is our main concern, so that or other materials which can be converted into simple sugars such as starch or cellulose that can be fermented further to produce the bioethanol that can be used in the gasoline engine. So depending on the bioethanol feedstock, there are different classified into three types. One is sucrose containing feedstock like sugar beet, sweet sorghum, and sugar cane. Other is a starchy material like wheat, corn, barley, and another one is the lignocellulosic biomass like straw, grasses, wood. So, depending on the availability of this kind of feedstock, we have already seen that this kind of sugar beet and this kind of category 1 and 2, this has the competition with the food cycle. So we are more focusing on the lignocellulosic biomass that is mostly available as the agricultural or forest residue. So those can be utilized for production of the bioethanol. So the major problem with the bioethanol production is the availability. That we have discussed that the availability of the raw material for the production over the year. So the availability of the feedstock bioethanol for bioethanol production can vary considerably from season to season and depending on the geographic locations as well. So this we can see that there are fourth generation of the biomass feedstock for bioethanol production. First generation is something that we are considering edible crops like starchy crops or sucrose containing feedstock. Second generation is the lignocellulosic biomass. Third generation is algal biomass, and fourth generation is some sort of industrial waste, or you can see some waste gas stream from the industrial output. Okay, so these are the four types of biomass feedstock depending on their generation and evolution. So we will be discussing in the further slides. So you can see this is one simple example that depend how this bioethanol production, that is the liter per ton of the sample or the feedstock, how it is differing. That you can see the maximum is happening for the rice. Rice is coming in the first generation biomass because it is having edible source of biomass. So it is giving more liter of bioethanol production, and uh, depending on the other biomass source variation, you can see most of the first generation biomass, they are having lot of potential to produce the bioethanol. But we are not focusing on the first generation bioethanol production because this can be competitive for the food cycle. So we will be generally moving towards the other generation so that our there will be separate production production area for or production field for bioethanol without affecting our food cycle. So you can see these we have segregate the generation wise bioethanol production and their advantage and challenges. Okay, so first generation, which is edible food crops like rice, wheat, barley and other things, sunflower oil, mustard oil, this thing. So these have the higher sugar concentration, so it will give the higher bioethanol yield. But the thing is, their feasibility of the production is still questionable due to competition with the food supply and also the land utilization, which increases the food cost. So we cannot produce lot of bioethanol in the industrial scale because that will be having limitation in the land utilization, as well as there will be subsequently the effect will be increase in the food supply, because we are using the edible food crops, so ultimately it is affecting our food supply. So this is not a feasible option, so we are moving forward with the second generation of bioethanol. So there we are having non-edible food crops like lignocellulosic biomass, like example wise forest residue, woody biomass, herbaceous biomass and municipal solid waste as well. So their advantage is they can grow on the poor quality marginal land, as well as they utilize very less water and fertilizer is required very less, because these are kind of residue and waste material we are utilizing. So this second generation bioethanol has lower greenhouse gas emission also compared to the other feedstock. But the thing is, it requires more capital cost due to the sophisticated processing equipment, and also it is not having that much of high sugar concentration. So lower energy density we are getting from the bioethanol, which has been derived from the second generation biomass feedstock, because it is comparatively lower energy density uh, than the first generation bioethanol. Coming to the third generation, we are focusing on the algal biomass. So they are cultivated on the marginal land with the water environment, land with the water environment, and there is low cost of cultivation as well, and there is higher conversion efficiency with higher energy density. So here the bioethanol produced from the algae is less stable due to um, their compositional variation, and this bioethanol produced from these algal biomass sources is comparatively less stable than the other kind of generation wise bioethanol production. Last one is the fourth generation, which is we are dealing with the waste based gaseous or solid feedstock using the genetically engineered microorganisms. So it is very advanced biotechnology has been used. So here we are minimizing the land and water uses compared to the first and second generation bioethanol production. Also we are maintaining here the circular economy, and we are producing carbon negative fuel. But the whole process takes lot of capital investment, because it needs higher pretreatment cost, as well as um, we are incorporating advanced genetically modified strains of the microorganism. So overall the process becomes costly. So we have to go for the optimum. Mostly we are going with the second generation biofuel and the third generation biofuel, because these are comparatively easy to go and with a higher yield of the bioethanol production. So, coming to the different routes for bioethanol production, so we are having sugar feedstock, starch feedstock, lignocellulosic feedstock, and algal feedstock, right? So, when we have sugar feedstock, it is having high sugar content, So we are directly extracting sugar juice from this milling, this pressing and cold pressing or hot pressing process. We are getting the sugar juice, and from the sugar juice we are going for the fermentation and produce the bioethanol. And when we are considering the starchy kind of feedstock, we are going either wet milling or dry milling, and then the milled product is going for the enzymatic hydrolysis. And when it is hydrolyzed and it forms simple sugars like C6, C5 to C6, then it is further taken for the fermentation and bioethanol is produced. Lignocellulosic feedstock, it has engagement of pre-treatment, which is having lot of cost involvement in this process, and lot of after pre-treatment we will be discussing what are the pre-treatment processes available. And then after the pre-treatment it goes for the enzymatic hydrolysis, which forms simple sugars, followed by the fermentation and bioethanol is produced. And other part of the lignocellulosic biomass feedstock, which follows the route of thermochemical conversion. It is converted by the gasification process. It is converted into syngas, and this syngas is followed by the catalytic conversion to produce the bioethanol in the thermochemical version route. And the last is the algal feedstock, which is directly is taken for the fermentation and produce the bioethanol. So we will be focusing more on the lignocellulosic feedstock of as biomass for bioethanol production. So the conversion of the cellulose and hemicellulose content to a simple sugars or monomeric sugar is harder to accomplish than the conversion of the starch directly, and presently used in the bioethanol production. So the main features for choosing a lignocellulose to bioethanol process is we have to go for efficient depolymerization technique for depolymerization of cellulose and hemicellulose to soluble sugars. We have to also ensure efficient fermentation for the mixed sugar C6 C5 sugars, uh, mixed sugar hydrolyzed, and to it 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 should be further fermented, as well as with their inhibitory compounds. Efficient fermentation should be happening, and we have to incorporate or integrate advanced processes to minimize the process energy demand. And lower lignin content of the feedstock should be encouraged to overall reduce the cost of bioethanol production. Now, the processing of the lignocellulosic component to bioethanol consists of four major unit operations. So we know that in the earlier of classes, when we discussed about the in one of the classes earlier, we discussed about lot of the pretreatment process and microbiological conversion processes we also already discussed. So here we will be not discussing so much in details, but just a touch upon the concept of bioethanol production through this major unit operation. So first one is the pre-treatment process. Then it follows the hydrolysis. Different hydrolysis processes are followed, and then fermentation, and last one is the product separation or distillation. So in the product separation also we are following the evaporation process, solvent liquid extraction process, and uh, those kind of existing liquid extraction processes are followed for product separation, like bioethanol separation from other acetone and ethanol, sorry, acetone and butanol, other products. We have to separate out the bioethanol. So you can see from this flow chart a simple idea how the bioethanol is produced in the process. So here the biomass is first, it is done for the pretreatment, where solubilization of the hemicellulose is being done. Then it is taken for the simultaneous saccharification and fermentation process, a well known process as SSF. There, enzymatic hydrolysis and fermentation are occurring simultaneously. Enzymatic hydrolysis, which convert the cellulose to simple sugars, and then it is simultaneously it is taken for the fermentation, where the conversion of the sugars in the presence of the various microorganisms, it is converted into bioethanol. Sugar to bioethanol conversion is taken place, and then the bioethanol is taken for the distillation and evaporation process, where bioethanol is taken out from the top of the distillation unit, and rest of the material is the filter wash to separate the lignin, because it is very difficult to decompose or convert the lignin component due to the high crystallinity of nature. And then the other are waste management, some of them process streams are being recirculated. So this is the overall a simplified diagram, process diagram, that how the bioethanol is separated from the biomass feedstock. So when we are considering the pretreatment process, so it aims to remove or alter some structural and compositional impediments to hydrolysis in order to improve the rate of enzymatic hydrolysis. Also, there will be increase in the yield of fermentable sugar from the cellulose or hemicellulose feedstock. So, this pretreatment can have significant implications on the configuration as well as efficiency of the rest of the process, and ultimately it will affect the economic. Pretreatment process has lot of impact on the overall economy. So a successful pretreatment must meet the following requirement, like improvement of the formation of sugars or availability of the subsequently forming sugars by the hydrolysis, avoid degradation or loss of the carbohydrate, avoid formation of the byproducts inhibitory to the process, to subsequent hydrolysis and fermentation process. By we need to optimize the process parameter to form such kind of inhibitory byproduct, and overall the pretreatment process should be cost effective. So you can see there are different pretreatment methods which are used for the disintegration of the lignocellulosic biomass. Chemical methods followed: dilute acid treatment, alkaline treatment, ionic liquid, organic solvents, and alkaline oxidative chemical treatments are followed. Mechanical: grinding, milling, chipping, microwave irradiation, sonications, these are some mechanical pretreatment. Also, it has stream explosion, hydrothermal, CO2 explosion, and ammonia, this ammonia inclusion technique. Also there are microbial conversion like fungi, bacteria. This kind of biological treatment are also followed. So here what is enzymatic saccharification, it is happening that enzyme which is help to uh, disintegrate the polysaccharides into monomers, and such monomers will be further taken into fermentation process, where the fermented product are is taken for the distillation, and from the distillation we are producing the pure bioethanol. This is another way to represent the schematic view of the process like this. The pre-treatment is just disintegrate overall the matrix of the lignocellulosic biomass, and then it is following this process like cellulose hemicellulose is converted into simple sugars by the hydrolysis process, and from the hydrolysis we are separating out methane, hydrogen, ethanol or butanol, and the lipid rich portion is followed by biodiesel production, and the lignin and the separate lignin component is taken for the high thermal conversion process like pyrolysis. From there we are getting either solid char or pyrolysis oil. So we have already discussed this biochar and bio oil in the last class, so we are not going for details in those topics. So here we have already discussed the other method for the thermochemical conversion like syngas, and the combustion wise it is having steam for the utilization of the rest of the residues. So this is the overall concept of how you are utilizing the lignocellulosic biomass to the production of the bioethanol as well as other byproducts. So this is the brief comparison between the pretreatment methods which is available for the bioethanol production. So physical methods are irradiation, milling, mechanical communication, and extrusions. So it is having lot of separate advantages and disadvantages, right? So we are not going in details. You can refer to the slide later on. So mostly we will be discussing about some chemical pretreatment methods like acidic and ionic liquids or basic sorry that basic solution treatment methods. So another one is the biological treatment which has the low inhibition rate, low lower yield of bioethanol, but it is also having low energy consumption and low requirement of the capital cost. So these are some comparison we already discussed in details in last few classes, I guess first of the two classes we already discussed in details about different existing pretreatment method. So we are moving forward, we can you can refer to the slide later on in details. So the pre-treatment methods, as it is finished, we are moving forward with the hydrolysis process. But the hydrolysis process, we are simply we are hydrolyzed into by the presence of water. We are hydrolyzed into simple soluble sugars. So the reaction is catalyzed by dilute acid or concentrated acid or either enzymes. This hydrolysis. So depending upon that, there are a lot of category of the hydrolysis process depending of the what catalyst you are using in the process, and they follow very mild conditions. When you are using the enzymes catalyst as a catalyst, it also give higher yield and maintenance cost is low comparative to the alkaline and acid hydrolysis. So this hydrolysis without a preceding pre-treatment method typically goes for typically yields of 25% yield, but when it comes after pre-treatment process, it can give greater than 90% of the bioethanol production. So the hydrolysis of the lignocellulosic biomass is more complicated than the pure cellulose due to the presence of the non-glucan components like lignin and hemicellulose. These are very difficult to decompose. So first one, the category, that is the acid hydrolysis, where different acid, mostly the sulfuric acids, are used for as a catalyst for the hydrolysis of the lignocellulosic content. So this acid hydrolysis mainly produce xylose from the xylene with cellulosic and lignin fractions remaining unaltered in the acid hydrolysis. This xylene is more susceptible to the hydrolysis by the mild acid treatment methods due to its amorphous structure compared to the cellulose, which needs severe treatment conditioning for its crystalline nature. So higher crystallinity, it will be requiring harsh conditions for hydrolysis. So the acid hydrolyzed substrates are then subjected to enzyme hydrolysis in the next step to give higher yield of the higher yield, like 100% yield reported for the corn stover and 90% yield of the bioethanol for the oak wood. So there are two basic types of the acid hydrolysis process, commonly known as dilute acid and concentrated acid. So you can see I have presented very briefly the difference between the dilute acid hydrolysis and concentrated acid hydrolysis. So in the dilute acid hydrolysis, we are using 1% sulfuric acid concentration in the process catalyst, and there are they are following two stage of the hydrolysis. First one, it follows low temperature hemicellulose depolymerization, and then it follows high temperature cellulose hydrolysis, where it has advantage like fast rate of reaction, facilitates continuous processing, but disadvantage, there is low sugar recovery, around 50% is limitation. Whereas concentrated acid, it is have using the 75% sulfuric acid with comparatively longer residence time than the dilute acid hydrolysis, and comparatively harsh conditions like 40 to 50 degrees Celsius, but there is complete and rapid conversion of the cellulose to glucose and hemicellulose to C5 sugars, and also it is contributing for the higher sugar recovery and less costly process. But the main problem is it is very difficult. 70% sulfuric acid, it is very difficult to work with like with this concentrated acid, and all acid must be recovered because otherwise that will be not be a cost effective process. So here you can see that two stage method for dilute acid hydrolysis. Lignocellulosic feedstock, it follows the dilute acid hydrolysis for the first stage, where the dilute acid hydrolysate is taken forward for the detoxification to remove the any inhibitory byproduct, and then it is taken for the fermentation to produce the bioethanol, and then the product is being separated. And the rest of the process which is not being depolymerized in the first step, the residual fraction is taken for the second step, dilute acid hydrolysis with higher temperature and higher residence time, and then the second distant hydrolysis is further taken for the detoxification and then followed by fermentation to produce the bioethanol. So this is the typical flow diagram for two-step dilute acid hydrolysis process, and this dilute acid hydrolysis process is mostly used in the industry. Second, it comes with the enzymatic hydrolysis. So it is the enzymatic hydrolysis of the natural lignocellulosic material. Is in nature it is very slow process because the cellulose hydrolysis is hindered by the structural parameters of the substrate like lignin and the hemicellulose content, also the surface area and the higher crystallinity of the cellulose. So utility cost of the enzymatic hydrolysis is low because we are not using any acid or alkaline catalyst for this hydrolysis, so it is cost effective in that sense, and also it is maintaining at a mild conditions like pH of 4.8 and temperature of only room temperature only, and does not have any corrosion problem. So these are some advantage of enzymatic hydrolysis. Now the enzymatic degradation of the solid cellulose is complicated process that takes place at the solid liquid phase boundary, where the enzymes are the mobile components. So when the cellulase enzyme which is applied for the this enzymatic hydrolysis process, it acts in vitro in on the insoluble cellulosic substrate. Three processes occur simultaneously. First is the chemical and physical changes in the residual solid phase cellulose. Next one is the primary hydrolysis is occurring, involving the release of the soluble intermediates from surface of reacting cellulose molecules. And then secondary hydrolysis is occurring, involving the hydrolysis of the soluble intermediates to further lower molecular weight intermediates, and ultimately it forms the glucose. So both bacteria and fungi can produce the cellulose enzyme for this enzymatic hydrolysis, and these microorganisms can be anaerobic, aerobic, mesophilic and thermophilic in nature. Next, it comes fermentation, which is the ultimate step to produce the bioethanol. So there are different categories of fermentation process. Uh, like separate hydrolysis and fermentation, pre-saccharification followed by simultaneous saccharification and fermentation. This kind of like separate hydrolysis and co-fermentation, consolidated bioprocessing, or simultaneous saccharification and co-fermentation. So these are widely used fermentation methods for the bioethanol production from the lignocellulosic biomass. So coming to the first process, which is separate hydrolysis and fermentation process, it is the enzyme where the enzymatic hydrolysis performed separately from the fermentation. So here the cellulose enzyme first convert the cellulose and hemicellulose into simple sugars, and in the next step the sugar rich hydrolysate is sent for the fermentation, where in the second step it is fermented the sugar into ethanol. So it is the process, like you can see here we are having separate hydrolysis and fermentation followed by combined sugar fermentation, because see, in the separate solid fermentation we are only processing the C6 sugars to bioethanol production, but in the combined sugar technology, sugar processing, we are going for the C6 and C5 sugars to form the bioethanol. So it is combined sugar fermentation is happening. So here you can see that lignocellulosic feedstock, its cellulosic fraction is taken, and enzymatic saccharification, by enzymatic saccharification you are getting C6 sugars, and pentose sugars is detoxifying and taken for the fermentation in the separate steps. But in the delignification with the alkali, and then it is mixture of hexose and pentose sugars are taken for the fermentation, so we are presenting combined sugar fermentation process. So in the next process, where the sugar from the pretreatment and the enzymatic hydrolysis steps are fermented by the bacteria, yeast and fungi, although the enzymatic hydrolysis and fermentation can be performed in the combined step. So that is called a simultaneous saccharification and fermentation process. So here it is often used in combined with the dilute acid or high temperature hot water pretreatment process. So this simultaneous saccharification and fermentation process, where cellulase and these enzymes convert the carbohydrate polymer into fermentable sugars, and these enzymes are notoriously susceptible to feedback inhibition or by the products like glucose, xylose, and other oligosaccharides. So these major advantages of this SSF are they increase the hydrolysis rate by conversion of the sugar that can inhibit the cellulose activity. They lower the enzyme requirement, higher productive, lower requirement for sterile conditions since the glucose is removed immediately from and the bioethanol is produced, and also the shorter processing time and lesser reactor volume. Then the main disadvantage is it lies different temperature region for saccharification and fermentation. Just an example, like low pH, less than 5, and high temperature, it may be favorable for our enzymatic hydrolysis, where the low pH can surely inhibit the lactic acid production, so it may adversely affect the fungal cell growth. So this optimum maintenance of the temperature and pH condition is some sort of limitation for this process. And more recently we are having more advantageous technology for where the simultaneous fermentation of the hexose and pentose is so-called co-fermentation is also happening, which is SSCF is referring, and the enzymatic hydrolysis releases hexose sugar, which can increase the rate of glycolysis such that the pentose sugars are fermented faster with the higher yield condition. And last one is the consolidated bioprocessing approach, where the enzymatic hydrolysis and the fermentation they takes place in the same reactor by employing a single strain. The celluloidic anaerobic thermophilic bacteria and the filamentous fungi, they are very potential microbes for this consolidated bioprocessing approach. So these are you can refer a detailed description of these processes. So I have only presented here some of the brief that how that fermentations occur in different categories that it has. So we will be now comparing the different fermentation strategies for the bioethanol production. So till now we have discussed different fermentation strategies. So uh, we have categorized depending on the difficulty and advantage of the process. So in the separate hydrolysis and fermentation process, we are separate optimal enzymatic hydrolysis and fermentation conditions, and difficulties or challenges have it needs pre-treatment. Separate reactors increases the overall cost because we are using two separate reactor. One is enzymatic hydrolysis reactor, another one it take for the fermentation fermenter. That two separate reactors increase the overall cost and higher cost for the hydrolysis, and some kind of in sometimes enzyme inhibition due to glucose accumulation. This also occurs in this process. When we go for the co-fermentation process with separate hydrolysis, which utilizes both C5 and C6 sugars to improve the process economy, which overall enhances the commercial bioethanol production. So but it has high investment cost due to use of the two separate reactor. The same process, same problem, just like, because still we are using separate methods, two methods, and the high cost of the hydrolytic enzyme and need potential fermentative microbes which can convert the mixture of C5 and C6 sugars. Simultaneous saccharification and fermentation process, where it has that lower cost and hydrolysis, fermentations are carried out in the same reactor, so it is beneficial in that sense. Also low enzyme inhibition is occurring due to simultaneous product we are separating out, and high ethanol yield and lower conversion time. So the difficulty is the different optimum conditions we have to maintain because we are doing it simultaneously for enzyme and fermented microorganisms. We have discussed in the last slide, like the pH and the temperature, though both should be in optimum for effective activity of the microorganisms. And also the higher cost of the hydrolytic enzyme still is a constraint, and higher medium viscosity, there might be limitation in the form of bioethanol production and only utilizing the C6 sugars in this case. So overall yield of the bioethanol uh, can be limitation due to the because it is only using the C6 sugars. And when it is part of this, that's combined fermentation technology, so we are utilizing C5 and C6 sugars in a combined way or mixture we are processing. So there are some limitation is lack of microbes for utilizing both of the sugars at a simultaneously, and pre-saccharification followed by simultaneous saccharification and fermentation process, which is having like produce the higher ethanol concentration single reactor for the hydrolysis and the fermentation. So it is having higher cost associated for the cellulose enzyme incorporation in the process, and also it is time consuming. In the last process where we are discussing about consolidated bioprocessing, so here we use the single reactor and the pretreatment is not necessary, so we are beneficial in that sense. Also we are using single microorganism for the fermentation and overall reduction in the cost. But also the main constant is the time. So time consuming, it is the process is time consuming, and there is lower bioethanol productivity. Here you can see some of the examples of feedstock depending on the hydrolytic enzymes it has been used. Fermented type, we have discussed lot of fermentation process. So depending upon that, these are the fermentation type has been used, and you can see this bioethanol yield depending upon that. So the highest bioethanol yield has been this pre-saccharification followed by simultaneous saccharification and fermentation. This followed a higher yield of the bioethanol, where they have used the olive tree for the feedstock. So it depends both on the hydrolytic enzyming, which hydrolytic enzyming you are using. Also depending on the fermentation process, the bioethanol yield will be fluctuate, like it will fluctuate the overall yield. So you have to choose the optimum, or the in a judicious way you have to choose the fermentation process for getting the maximum bioethanol yield. So now we will be moving forward with the biobutanol production. So bioethanol, we that we have discussed that it can be used, no doubt it can be used in the blend condition with the diesel engine, diesel fuel, and there are a lot of improvement with the advanced technology that we can do for the production of for advanced production of the bioethanol, so that we can take that bio-based ethanol fuel in the market in a sustainable way, right? Similarly, when we talk about the biobutanol production, it is also in a similar characteristics of the gasoline, and we can also replace that the gasoline consumption with the biobutanol. Excuse me for the 2 minutes. I'll be back after 2 minutes. So biobutanol, which is introduced as a organic alcohol that can be utilized as a biofuel or fossil fuel extender. Extender means we are utilizing in a blending form with the existing fossil fuel. That's why it is calling extender. So due to its reduced volatility when compared to the other biofuels, biobutanol ig- is said as an example of superior renewable fuel. So biobutanol is nothing but four carbon molecules with the presence of the OH group attached. So here it is like colorless liquid fuel having flammable characteristics that can be burned efficiently in the presence of oxygen. Biobutanol owing significant properties such as high energy content, hydrophobicity, blending ability and compatibility to the combustion existing engines, corrosion and octane rating, higher octane rating, is increasingly regarded as a renewable source of energy. So it has the ability to blend with the gasoline ratio up to 85%, just like bioethanol. So blending of the biobutanol with the diesel could reduce the shoot emission or shoot formation due to the presence of the oxygen in the butanol molecule, as well as improvement in the fuel mixing process due to the longer ignition delay. So, butanol can also be used safely and advantageously to the higher blending ratio with the diesel fuel in the existing diesel engine, as it could provide the higher or good thermal efficiency and reduce the emission. So, biobutanol has analogous characteristics with the gasoline. The property of the biobutanol, a density 810 kg per meter cube, energy content around 23.4 megajoule per liter, which has the similar energy density to the petroleum and 25% more energy per liter than the ethanol. So you can see that higher energy content giving it more miles per gallon than the gasoline. So the synthetic yield of the biobutanol, which is 0.42 weight percent from the corn stover, having 90% cellulose content. This is an example. They are very promising source to be utilized as a biofuel. So this butanol is crucial, also a crucial element because it is widely used as industrial solvent and in virtually manufactured either by oxo process of the propylene or aldo process of the acetaldehyde. So it is also used in the um, also can be manufactured using the chemical processes. Industrial synthesis of the biobutanol was first developed or achieved by anaerobic ABA fermentation, which is acetone, butanol, and ethanol fermentation of the sugar components like molasses and cereal grains using Clostridium acetobutylicum. That is Weizmann's organism. It is known as Weizmann organisms. It exhibit higher yield of the biobutanol. So this ABA fermentation was the second largest industrial fermentation process after ethanol, due to the economic importance of the acetone as well as butanol prior to the introduction of the petrochemical solvents. So for the synthesis of the biobutanol, lignocellulosic biomass typically has should have cellulose content around 34 to 47%, hemicellulose content around 19 to 27%, and lignin content around 5 to 4%. Here you can see some of the strains which have been used to produce the butanol in this ABA fermentation process. So here most of the strains are Clostridium species, and different strains are used. Raw material are mostly glucose, and some of the carbon monoxide is also used. So butanol production is higher for this glucose using this particular strain of the Clostridium. So we are having the main product in all of the cases, mostly butanol, acetone, and ethanol. Some of the process they produce isopropyl alcohol, isobutanol, methyl butanol, and this kind of other byproducts. So we will be now discussing that how this biobutanol is superior to be used than petroleum diesel, and even it is superior than the biodiesel. From the comparative analysis we can see. So this butanol diesel, this blend we can use in an eco-friendly manner, and this increasing the butanol blending in the diesel, this can improve the emission reduction from an engine. So biobutanol production, which is recommended as a more cost-effective method than the production of the biodiesel. You can see from this comparison that biobutanol, which is produced from the microalgae biomass mostly, and this is crude oil, the main thing, and the anaerobic fermentation is followed for producing of the biobutanol, where fractional distillation, high temperature, high pressure conditions are followed. This is also cost intensive. Here, biobutanol production, we are getting other useful products like acetone, ethanol, methane gas as a fermentation of the spent biomasses. So here we are not getting any byproduct. We are getting the distillated product, nothing like byproduct, less like fermentation process. So biobutanol production, in this case we are cost is higher, but cost is higher in this sense because micro algae biomass, we are maintaining the anaerobic conditions for the fermentation and the distillation process. The overall cost of overall processed cost analysis, by the cost analysis we can say that this is costlier, and CO2 emission, obviously it is lower, and effective energy consumption is lower when it is compared with the biobutanol and biodiesel. Then most of the cases we are having that this process is only having microalgae biomass as a carbohydrate source, where the biodiesel, it also needs the methanol as a external source to convert the biomass into biodiesel, because we need ethanol. So here we also need the catalyst, and transesterification process is followed. Here we only get the glycerol as a byproduct, but here we get other alcohols like bioethanol or bioacetone methane gas for the byproduct, which is a beneficial process for biobutanol production. Also the energy consumption is lower because separation of the surfactant and the separation of the ABA, that acetone, biobutanol, and the these other ethanol, these components are separation, this overall energy consumption is lower. Whereas biodiesel, it follows the pre-treatment process, lipid extraction process, and transesterification process. So there is overall higher energy consumption in the biodiesel comparative to the biobutanol. So overall we can say that biobutanol production is more effective than the um, biodiesel production, and it can be efficiently, it can be blended with the um, it can be blended with the diesel fuels or the gasoline fuels in a good proportionate way, like 85% weight percent. So this biobutanol production is also similar to the bioethanol production, which having primarily four steps for butanol production using this Clostridium strains of microorganism. So the pretreatment process, it follows physical, chemical and biological. Also, syngas does not require pretreatment process before the fermentation process. Second step is the hydrolysis, which breaks down the complex molecule into fermentable sugars, which can be followed acid, alkaline, and enzymatic hydrolysis. And this fermentation process, we already discussed this kind of variations of the processes which metabolize the fermentable sugar to produce the biobutanol. And last of all, we are going for the distillation for the product separation in from the mixture of fermentation broth or other mixture. The similar process is followed in the biobutanol, just like bioethanol. So main is the ABA fermentation, which forms these fermentable sugar into this category of three solvents like acetone, butanol, and ethanol. So we have to, and some acids are also produced, acetic acid and butyric acid. Gases it produce carbon dioxide and hydrogen. So we can separate out the biobutanol from this process using the distillations and other separation process available. So so here you can see this starch biomass which follows gelatinization and hydrolysis to form the fermentable sugar. So here the advantages is simple and fast saccharification process because we are using the starch biomass. Also cheap amylase and high sugar conversion yield because it comes under the first generation fuel biomass for the biofuel production, and there is no need of the pre-treatment and no inhibitory compound is present, and efficiently we can go for the direct fermentation method to form the butanol. But the problem is limited sources of the starch waste, and also there is huge amount of the water is present. So that may be two limitation of the starch biomass, but overall it is good source of the high sugar conversion yield. In case of the lignocellulosic biomass, it needs the pretreatment as well as hydrolysis, then we get the fermentable sugar. So it is most abundant material, one of the plus point, but this has other lot of difficulties like mostly it requires harsh pretreatment process like acid and alkali pretreatment, and expensive cellulose and low sugar conversion yield we have observed or reported, and presence of inhibitory compounds like more concentration of the cellulose, it forms as a inhibitory in the process. So saccharification produce mixture of the sugar, so C5 C6 sugars conversion is little bit difficult than only conversion of the C6 sugar, and there is insufficient direct fermentation of the butanol. Coming to the algal biomass, so we are algal biomass is obviously it is having good simple and fastest growth. Also we are going for the CO2 sequestration in the form of algal biomass growth. So carbon dioxide fixation is also very important approach for the algal biomass growth, and not competitive for the land and food crop utilization. And there is higher composed of the lipid and carbohydrates, so this lipid portion we want to extract so that we can produce the biodiesel out of that, and these carbohydrates are further taken for the pretreatment and hydrolyzed to form the fermentable sugar. So but the thing is, there is higher cost of the harvesting and then drying and extraction. Additional process is required to ferment the algae, high capital cost investment, and there is low biomass concentration. So ultimately, when we get the fermentable sugar, then we follow for the ABA fermentation process. So ABA fermentation process typically follow batch fermentation, or fed batch fermentation, and continuous process fermentation. So here, batch process, if we get the low sugar concentration, and for the continuous process, mixing of the solventaneous and acetogenous, these two methods of the fermentation, ABA fermentation, we will be discussing in the next slide. And there is low sugar consumption rate also. So ultimately, we are getting these three component, that is the ABA fermentation, that is acetone, butanol, ethanol as a solvent, and we need to go for the further distillation to separate the product. Now we will be discussing that how this we can enhance the biobutanol production through the different pretreatment technology available. So we have to go for the depending on the biomass feedstock characteristic. We have to choose the appropriate pretreatment method to enhance the biobutanol production, right? So coming to the acid treatment process, here we solubilize the hemicellulose and condensation and precipitation of the solubilized lignin. So it is very low cost, and but toxicity by product is very high. So it prevents the synthesis of the inhibitor, and dilute acid is employed. So here mostly we use the dilute acid process. We have discussed few slides back that concentrated acid are very difficult to handle in a industrial level. That 70% concentrated sulfuric acid is very we have to be very cautious, and we this much of concentrated acid should be sufficient enough to go for the recovery of those acid, because otherwise there will be um, lot of economical lacking we will be having, because we will lose lot of acid, concentrated acid. Coming to the ammonia fiber expansion process, so it is less effective for the biomass with higher lignin content. So it increase the, so this kind of um pre-treatment process that we have discussed here in a detailed way. So here mostly it represents the most available technologies for the pre-treatment of the lignocellulosic biomass. But most focusedly, when we talk about this a milling process, which is used widely for the bioethanol and the biobutanol production. So you can refer to this detailed pre-treatment process, because it is nothing but as a general description for efficient pre-treatment process, just like we follow for the bioethanol production. The same kind of pre-treatment process also we follow for the biobutanol production, depending on the technologies available. For the most and most significant step is the ABA fermentation process for producing the biobutanol. Whereas we do the fermentation, the microbial fermentation process for the bioethanol production. That is the main uh, difference. Like, so now we will be discussing on the of ABA fermentation. So this ABA fermentation, we have to maintain the medium for the fermentation, which is yeast extract and pH should be maintained at 5.5 to 6, and this inoculum is maintained 10% volume by volume percent. So here by the fermented sugar we are maintaining a sterile condition, perfectly anaerobic conditions for with nitrogen. Nitrogen in an atmosphere, 150 rpm agitation speed. This is also important factor as a process parameter for the biobutanol production. Also the maintained temperature for higher yield of the biobutanol is 35 to 37 degree, and it requires 72 hours of duration to produce the biobutanol out of these fermentable sugars. So we are mainly getting the gaseous acids and solvents, so we are separating out the solvents. So this process is called so-called acetone butanol and ethanol fermentation, which is involves the basic biphasic fermentation stage, which are acidogenesis because acid also produced, and solvenogenesis because solvents are also produced in this stage. So these microorganisms produce solvents like acetone, butanol, and ethanol, and acid like acetic and butyric acid. Also gases hydrogen and carbon dioxide as its byproducts. So these multiple byproducts are derived from this one fermentation process, which constrains the biobutanol yield, concentration, and productivity. So the cells can only stand solvent concentration less than 2%, otherwise they will inhibit the culture of the cells. Beside the biphasic fermentation involving two distinct phases make the fermentation process more complex, because we are incorporating this in this process we are getting liquid phase and gaseous phase also. So this biphasic fermentation, it makes the fermentation overall process very tedious and complex. So these stages are critically affected by the pH conditions, intra and extracellular environment, like this perfectly anaerobic conditions and temperature pH condition. So the when the pH value of greater than 6, it can cause the cells to produce more acid. So our objective will be hampered for that case when we maintain the pH greater than 6, and it sees the solventogenic phase to produce more solvents. So the ABA fermentation from the biomass also, it requires multiple processing steps, making the whole system more complicated and tedious. So the biomass needs to be hydrolyzed into simple monomers before being consumed by the cells for biobutanol production. So we have to this fermentable sugar we need to convert. The sugar concentration should be adequate enough to induce the solventogenic phase and prevent the other substrate formation. Most of the lignocellulosic biomass they require for pretreatment process, and which can be good enough for enzymatic action. Some biomass hydrolyzed is required for the detoxification process to remove the inhibitory components present after that pretreatment and the hydrolysis processes. So all of this factor can reduce the biobutanol yield and productivity, and subsequently they make the recovery process from the fermentation broth inefficient. So we need to optimize the process to maximize the biobutanol yield, so that the recovery process becomes easier. If the biobutanol percentage is very lower, then the distillation and other processes will be very inefficient to complete extraction of the biobutanol from that biphasic solvent phase. So when we talk about the biobutanol biorefinery, this is a good concept. So depending on the biomass fractions, we can see that how the products and uses we can refer. So the cellulose biomass product is acetone, butanol, and ethanol. Also the gaseous and other fuels are the chemical. So it is having a lot of other compounds like HFM, HMF, that is the hydromethyl paraldehyde. So this kind of component is used in the food industry. Lot of uses are there, and solvents and lubricants, chemical polymers is used from the levulinic acid. Hemicellulose, which having pentose and xylose, this is also useful for butanol acetone production. So depending on the biomass fractions variation, that extractive lignin, hemicellulose, cellulose, we can build a biorefinery effective for various production of the chemicals which have various uses in the chemical industry as well as food industry, also different solvents and polymer industry. You can see there are some uses like agricultural dispersant and emulsifier. Also there are medical applications also in the extractive sense. So we can form a good depending on the biobutanol production, we can also build up a good field of the chemicals for the biorefinery. Last topic of the discussion for today's session, that is the biotechnological advancement for the biobutanol production. So it is having the genetically advanced different strains of the microorganism that can enhance the biobutanol production. So the prevalent substrate in the ABA fermentation is the lignocellulose sourced from the agricultural waste mostly. Nevertheless, the primary challenge associated with the lignocellulosic substrate lies in their limited fermentable sugar release from for the microorganism. So there are biotechnological methods that have improved the butanol production efficiency by optimizing the strain performances, and strain development for the biobutanol production has focused on aspects like butanol tolerance. We have to improve the substrate utilization, increase the butanol selectivity and selectivity and productivity in the lignocellulosic biomass waste. So there is various genetic approaches such as chemical mutagenesis or genome library construction, which can enhance the microbial resistance to the butanol. So if the butanol concentration is beyond suppose 10 beer supports 10% in the froth or the fermentation mixture, then the cell cannot be grow further. So we are genetically modifying the genes of the microorganism so that it can resist more concentration of the butanol, and that also effective for the higher yield of the biobutanol. Suppose an example, overexpression of the genes like GROW-ISL has been shown to significantly increase the butanol tolerance and production in the acetobutylicum. This enhances butanol concentration, which is a key focus in the biobutanol production. Right now, the genetic engineering has succeeded in increasing the levels of the butanol production. Suppose an example: upon inactivating the acetone decarboxylase gene, the C-acetobutylicum has shown a notable reduction in the acetone level and significant increase in the butanol ratio in the ABA fermentation. So, which is very beneficial for the overall increment of the yield of the biobutanol production. So, such way, suppose overexpression of the acetaldehyde or alcohol dehydrogenase gene, which can further elevate the butanol output. So overall productivity will increase. So thus way, by the help of the genetic engineering, we can modify or we can tune the genes of the uh, genes of the microorganism to overall effective higher yield of the biobutanol production. Additionally, when algae are immersed as an alternative substrate for the biobutanol production, microalgae can increase their carbohydrate content through pathway engineering and diverting the flow towards the carbohydrate metabolism rather than lipid production. So in such way, when there will be higher carbohydrate content in the algae rather than lipid content, so there will be higher conversion of the carbohydrate to the biobutanol production. So there will be biodiesel production will be suppressed. So there are also other promising approaches for increasing the butanol production. They include redox metabolism to enhance the coenzyme availability using the redox mediator and integrating the bioelectrochemistry in the bioreactor. We can overall increase the yield of the biobutanol production. Uh, is there any question from today's class that we can discuss for the rest of the 5 minutes? In the next class, we will be discussing about hydrogen, methane, and other methanol production from the biomass sources. This hydrogen production is one of the very interesting topic for biomass, because hydrogen having lot of application as a good energy source. So we will be discussing lot of interesting sources and applications in the next class. So, is there any question? Okay, so if there is no other question, I would like to end this session here. Thank you for joining.